So we're going to begin our discussion here. Proteins as the next macromolecule. And when we say macro, these proteins are really macro. These proteins are humongous. These are gigantic molecules compared to, a glucose or even a starch. I think the proteins would probably outdo some of the starches in terms of size. But the difference is that the starches tend to be sort of stretched out linearly, like in long lines, whereas proteins are more blobby. Okay, so what is a protein? Protein is also a polymer. Remember what that word means, right? A polymer. Just like carbohydrates, like starches, are made by putting sugars together, right? We're going to take certain molecules and stick them together in repeating patterns to make proteins. But a protein is a polymer of molecules called amino acids. So the first thing we have to do is talk about what amino acids look like. And in your textbook on page 40, there's a nice chart that shows 20 or so amino acids that are essential to humans. There are many amino acids, but in terms of biology, there's only some that actually are critically important, it seems. So if you look on that page right now, you'll see that I think there's 20 on there that are shown. And these are the 20 amino acids that are found in the proteins in living things. Okay? All right. So what is an amino acid? Well, you can tell by its name that it must have some functional groups. And of course, an amino acid has the amino group, and it also has a carboxylic acid group, or the carboxyl group. And so the simplest formula for an amino acid is the following. Oh, let's go down. We'll use an R to represent a bunch of stuff stuck to the top. And then that R is attached to a sort of a carbon atom here, which has, now remember, this is in 3D, this is the tetrahedral shape. Can I go up a bit? Yeah, 1 second. Okay. Remember, it's a tetrahedral shape. So I'm going to draw it sort of like this, 1, 2, 3, but you have to remember that those are sort of sticking in and out of the board a little bit, right? Think of it like the model that we have there. And so what you have is you have a carboxyl group on one side, COO. Your textbook puts it in the ionic form with the H missing. So I'll draw it the same way as your textbook so it doesn't confuse you. But you could put the H on there too, right? COOH. And then when something joined with it, it would attach there. Actually, you know what? Because we're going to be attaching these later, perhaps we should. I'm going to draw it in its regular form, not the ionic form. COOH, like that. And then over here, we would have the amino group, the NH2. We could draw it this way. There's the amino functional group. But it's more convenient to draw it as just NH2. Okay? And you have to remember that NH2, whoops, that's not NH2. You have to remember that NH2, the amino group, can attract another hydrogen and become NH3 plus, right? It's the one that forms a positive. And then there's a hydrogen on the bottom. So all amino acids have the amino group like this. They all have the carboxylic acid group, and then they all have a little hydrogen filling out the last bond. The only thing that's different is whatever's on top, whatever the R stands for. And that's what makes the difference between all the different amino acids. So we won't draw them all, but we'll draw a couple sort of examples just to get the idea. We all good? Okay. So here are some selected examples. The simplest amino acid is glycine. Glycine is the standard amino acid. COOH, NH2, amino group, carboxylic acid group, and then on top it just has a hydrogen. That's what the R stands for this one, which is glycine. Most of them end with -ene, lysine, leucine, so on, but there's a couple that don't. Remember, there's always exceptions to the naming systems. Unfortunately, as things get discovered, we sometimes come up with the realization that there's a whole family of molecules much later. And so some of the names don't fit that picture. Let's see. Alanine is an amino acid that has NH2, and instead of a hydrogen, it has a CH3, which we commonly call a methyl group, because it's like a little methane sort of stuck on the top, right? Methyl group. So that one's called alanine. Some of them have quite complex structures on top. So I'll draw one of the more complex ones, or a couple of them, as long as you're with me. We'll go down a little further, because these guys, so when you draw these guys, leave lots of room on top, okay? So leave lots of space up on top, because the bottom part, C, H, COOH, alanine was the last one, NH2. So this particular one I'm going to draw has a CH2, and then attached to that, it has this nice hexagonal carbon structure. So remember, all the corners are carbon atoms, but we're not showing them for simplicity. And then there are these double bonds that between some of those carbons, like this. And this one is called phenylalanine. Phenylalanine. And the reason for that name is if you go back to alanine, see the CH3 on top? This one has sort of the same thing, except it has this extra little thing added on top. This hexagonal thing, and of course that's where it gets the name phenylalanine, because this is like a phenyl, this little group that's on top. So phenylalanine. Phenylalanine. And we'll draw one more. So there are some that have, maybe one or two or three of these. Can't think of one with three, but there's definitely, I think it's tryptophan, has two hexagonal things stuck together. The last one we'll draw will be this big one, COOH, NH2. And of course, we will not be memorizing all these structures. We just want to be able to identify them. So this one has CH2, which is a shortcut for drawing a C with two H's sticking out of it, right? And then it has another CH2. And then it has another CH2. So there are three CH2s going up. Then there's an N with an H on it. And then there's another C. Uh-oh, I hope I don't run out of room. There's another C. And this one then has an N and an H2 stuck up here. And then there's a double bond over here to an NH3 plus, and so on. So that's a very complicated one, right? Very complicated. It's one of the bigger ones. Arginine. Can I scroll up? Sure I can. All right, so this is NH2. We corrected that because we noticed that it was not NH3. It's supposed to be NH2 plus. The double bond of the carbon means that one of those spots for an H is gone, right? It's being used up. So it has to be NH2 with a plus sign on it, because nitrogen has three hands, basically. Two of them are attached to the carbon. So if there's an extra H, that would be four hands. It must be attached to one of those unshared pairs on nitrogen, the coordinate, the covalent bond. So that's good. We've corrected that. All right. What we have to be aware of is simply this part. This is how you tell that something's an amino acid. They all have this thing on the bottom. So that's all you have to really learn to recognize. You don't have to remember all the top parts. And it's just an amino group carboxyl group and an H. Oh, speaking of H, look what we did. I forgot the H right here. Oh, stop doing that. I don't know why my smartboard is not very smart. Right here, we forgot a little H. Let's put that in as well. They all have that same pattern. Okay, so far so good. So now what we have to do is we have to figure out how they join together. Okay, so let's draw two of them. Okay, let's stick a couple of these together. Let's take two amino acids. So what I'm going to do is I'm going to draw them slightly differently so you can see the bond. So let's take this bottom, oh shoot. That's not supposed to do that. Okay, we corrected the problem of using markers on the smartboard. All right, so we're going to draw an amino acid. So I'm going to put the C here, the central C, with the H on the bottom. And then over here we have the NH2. So I'll put there's the amino group on the end. And then over here, what we should have is a C, double bond O, right? And then an OH. So instead of writing it as just COOH, I'm drawing the whole structure now. That's the same as this, right? Just a shortcut. This is a shortcut version. So we want to see the whole thing so we can see just where the bonds are happening. So far, so good. And then up on top, I'm going to just kind of draw it in blue because it doesn't really change much, is the R, whatever's hanging on up there, whether it's the hexagon shape or just the hydrogen or whatever. We don't need to draw that every time because it doesn't matter because it's not involved in the bonding of the two together. Okay, and then beside it, we'll draw another one. We'll draw, I'm going to erase, because I just ran out of space, I'm just going to erase this so we have room. Okay. All right, let's draw an N here. And then that N will have an H on it. And that N will actually have another H on it. Oh, I didn't leave enough room, did I? Got to move over just a tad. So here's the N, H, H. Up another one, which will be attached to a central carbon, right? And it'll have its little H down here, and it'll have its little COOH over here, and it will also have some other R. So I'll call this R2 and this R1 to show they're different Rs. They're two different amino acids. Now, they could be the same one, too. The same amino acid could join to itself in a chain. Now, if you look at the middle here, what do you think is going to happen here? Knowing what you know about how things sort of bond, these two functional groups, this amino group and this hydroxyl group, are going to bond or interact with each other. Look what's available there. Yeah, there's an H and an H and an O, which makes us think that this could be a dehydration. If we pull the water out, we could make the bond. So let's do that. Let's pull the water out and show what it would look like if I take this OH and this H and I remove it from here, like a dehydration. You can see how it would stick together. Okay? And so what it would be the original amino acid on this side, N, H, H, and then I'll put its R in a minute. And you'll see that the C of this one, right, will be the C of the carboxylic acid group is still there, and the double bond O, that's not involved, but the OH is gone, and it's attached directly to the N of the other one. Its H is gone. So we have this situation. I think I've got that. And I'll put the R's in. The R's just, R1 here is carried along. Oh, I meant to do that in blue, but didn't work. And the R2 that's stuck up here is carried along. And so we have this interesting little bond situation here in the middle. This CN bond right here. And it has a name. It's called a peptide bond. Carbon-nitrogen bond between these amino acids. The peptide bond. Now you can also notice that we could just go over here and add another amino acid and do the same thing, right? And just keep on going. So we could have 2, 4, 10, 100, 5,000, as many as we wanted, stuck together in a line. And so when you do this, you create a very long chain. So this, you create a long chain of amino acids like this. I sure can go back. So this long chain of amino acids creates what is called the primary structure of a protein. The primary structure of a protein. But it turns out that's not the end of the story. The protein is a bit more complicated than just that. So far, what we have is basically a bunch of amino acids stuck together with peptide bonds. Does anybody remember from grade 11 what the enzyme for breaking down proteins in the stomach was called? Pepsin, because it breaks the peptide bond. Just a little aside there. So you see, all these names do make sense after a while. So this could go on, and this chain could be hundreds of amino acids long. Literally, yes. Can I go up a little bit? Yes, I'll go back. I won't stop recording this time. I'll just talk while you're writing that down. So that's the primary structure. Okay. Now, the interesting thing is that because the amino acids are all slightly different on top, they all have different R groups, that means there are different molecules sort of attached up there, which means they all have little different pushes and pulls. They're not all the same. So the tryptophan molecule pushes differently than the alanine on each other when you put those together, which causes the chain to not stay in a nice long even line. It causes it to kink and bend and twist. And so what happens is the kinking and bending and twisting of this line basically produces what we call the secondary structure of a protein. They fold into patterns, okay? I'm just going to simplify. I'm trying to figure out how to draw these two because one is very difficult to draw, but I think we can do it. So instead of this picture that I've drawn here, what we tend to get instead is a kinked up picture, which kind of tends to spiral. It's difficult to draw that, but it's basically a spiral-shaped line that's something like this. And it's because each of the little R groups on these amino acids is pushing and pulling on each other. You'll notice that some of those R groups had ionic ends, like the H2+. And those ionic ends have strong push and pull forces. And so it causes this to either kink up in, the two most common ways it kinks are the spiral shape, or it's possible that if I go back to my picture, okay, it's possible that another chain could form underneath this chain, attracted very lightly by what we call hydrogen bonds, which are essentially the water dance, the pushing and pulling. This oxygen right here, for instance, if I just draw it, has two unshared electrons right there. And so another amino acid, the hydrogen of, say, another amino acid like this, for instance, might come up above here, if this was another line of amino acids, and be slightly attracted to this negative spot. And it just kind of holds it there. So what you tend to get then are two chains, or three or four chains, or several chains that are, instead of spiraled, they're sort of lining up like this and forming almost like a sheet of paper. But they don't form flat. What they actually do is they bend. So this is the part that's very hard to draw. I'm going to try to get a drawing here to show this to you. So it bends like this. So imagine a piece of paper with folds in it. So there's my piece of paper, right? And it's folded. To give you the fold 3D effect, I think if I do some shading here, let's see what happens. Line color, crayon. Let's see if I can make this work. If we sort of make this, oh, that's the wrong color now. If we sort of shade in this crease to make it look like it's dark in the middle, but not so dark on the ends, like that, but darker sort of here. I'm going to do some art here. And then this one's very light. This one would be very dark. It would help if I had drawn this more evenly. Let me fix that first. Line, pen. See, it's not listening to me. There it goes, pen. So this goes up, and then down, and then up like that. Okay, crayon, green. So this is a dark, right? And then lighter up here. Doesn't work very well with smartboard, but the idea is that these are folds that are away from you, right? And these are, so it's basically going in and out of the board, folded like a bent piece of paper. What I can do is pause, and I'll just bend a piece of paper and show you. Okay, so this is called the pleated. Oh, let's get the crayon out of here, back to the pen, go back to our color. This is called the pleated sheet form. And these are the two most common ways that these lines or chains crumple up. And this is called the secondary structure of a protein. So not only is it a long chain, but the chain itself has a specific shape. It's either a spiral or a bunch of chains making a pleated sheet. So far, so good. Okay. Now, what can happen is what's called the tertiary structure. There's a third thing that happens. This happens because of the R groups again. The R groups are interfering with each other, and so instead of just a spiral line like this R group right here attracts to this R group right here and causes the spiral to sort of kink on itself, like that. This gets pulled around. And so what happens is the spiral then begins to kink and twist, where different parts of it attract to other parts because of the different R groups. And the spiral produces this big, tangly, globby mess. The best way to visualize a protein is to think about a big ball of string, which I left in the other room. So I'm going to go and grab it. Well, not recording. So the tertiary structure, which looks like that ball, like a spring all wound up, is caused again by interactions of the R groups. But in particular, you'll notice if you look in your book, we didn't draw any, but in particular, there are some amino acids, in particular, there are sulfur atoms. Sulfur atoms found in a few of the amino acids, when they bend and kink around, the sulfur atoms bond or hold themselves together, creating what are called disulfide bridges. Disulfide, I'm writing too fast. Disulfide bridges. So these are the bonds between the sulfur atoms that are more responsible for twisting up the coil into this tertiary structure. Tertiary, T-E-R, tertiary, which means the third. That's a word that means third. Primary, secondary, tertiary. Okay? So there's the long string. The long string is the primary structure. Then it gets coiled or pleated in a sheet. That's the secondary structure. And that happens because of interactions between the R groups, but more so because of charge differences. Okay, so that twisting and coiling is more because of the pushing and pulling of electron pairs in the R groups that are like the Vesper theory, trying to get away from each other. But this tertiary structure, where the whole coiled up spring actually turns on itself and creates a tangled up web or ball or knot, that's caused by the sulfurs inside some of these that are actually grabbing onto each other with a fairly strong bond called a disulfide bridge when they stick together. Okay? All right. So, one last thing. One last thing. To make it worse, what can happen is you can have one ball of string. So I'm going to draw a line here because now we're on what's called the quaternary structure, which is quaternary, which is the fourth level of structure in a protein. And that is, you get one coiled up spring. Here it is, right? La 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 la, and just this is fun to draw, so I'll just draw it all around here. There's the tertiary structure of one protein, or one string. And remember, this is 3D, so it's lumpy and bumpy. And what can sometimes happen is another one will become sort of associated with it and attracted to it. So you can actually get two blobby knots that are stuck together, like this. So two tertiary, I'm going to call them knots, of protein or of amino acids can be attracted together. And again, it's all, this attraction is all because of all the pushing and pulling and what's going on with all those R groups that are attached to the amino acids. So this makes the quaternary structure. So this is the final picture of a protein. So we'll call this a protein. Yay. So I think what we'll do is we'll just do a quick summary of this. A very quick summary. Once you're finished drawing and writing. Summary. Let's summarize then. Whoops, let's go down here. All right. So a protein is a complex molecule. It has a primary structure. And the primary structure is simply the chain of amino acids. I'm going to put AA for amino acids, linked by peptide bonds. Linked by the peptide bond. That's what we call the primary structure. The secondary structure is describing what happens to that chain. The chain becomes kinked. Due to the attractions of polar, not paler, polar, of polar or charged regions. Of the R groups. Remember what the R groups are. The R groups on the amino acids cause kinking and bending. And we see two specific sort of types of this. Okay? I realize you're trying to catch up to me. So we get the spiral structure is common, and the other common structure is the pleated sheet structure, both of which are commonly found. That's the secondary structure. The next structure is the tertiary structure, which means an additional bending and folding and kinking of the spiral or the sheet upon itself, which is due primarily to the disulfide bridges. Okay, so now we go like this, and we have what's called the tertiary structure. The tertiary structure, the third level of structure, is where the spiral or the sheet bends and folds on itself due to attractions, particularly though the attraction that seems to be responsible for this, particularly the disulfide bridge. Okay, the disulfide bridge, which literally forms a bond that holds it into, it forms a bond that holds it into this particular arrangement. Bends and folds on itself due to attractions, particularly the disulfide bridge. The disulfide bridge is an actual bond, so it's fairly strong, between sulfur atoms my line's a bit too thick for writing. Bond between sulfur atoms on the R groups. When I write, I should use a thinner pen. I'll try to remember that next time. It's good for pictures, but it's hard to read. Actual bonds with an S, between sulfur atoms on the R groups. So there we go, we have a tertiary structure. So far, so good. Now, there's even another layer called the quaternary, the fourth layer of structure. And that is two, it could be more than two. I'm going to say two or more. Usually it's two or more tertiary, two or more tertiary I'm going to call them knots because that's what they look like, right? They're like twisted chains. Two or more tertiary knots linked together in a complex tangle. And that's the fourth level of structure for proteins. And we should take a few minutes and just talk about what this means in terms of the function of proteins and what they can do because of this complicated structure. Remember, all of this complex structure is simply due to the attraction and repulsion between charged regions of the molecule. And that's all because of the way the atoms are trying to fill their outer orbits. the filling their outer orbits causes distributions of charge that we call polar regions, and it causes bonds. And those things are all working together. So in other words, if you put amino acids in a cup and just shake them up a little bit, they will do all of this automatically in response to that one instruction. They just do it. Nothing has to make them do it. It's a consequence of one simple rule. Okay, so let's talk about the function of proteins then. What can they do? Well, they can do, because they're so complicated, they can do lots of things. Lots of things. They can function mainly, what proteins do is they function as structural components. They can help form more complex structures of cells. Because they are a tangly knot, and because that tangly knot can be manipulated and twisted and take on different shapes, they can have situations where being near other molecules causes them to open a hole in the middle. So they can function as gates or doorways on cell membranes. And that's a very important function. We'll talk about that later. They can also function as enzymes, which we're going to talk about briefly in more detail in a day or two. Enzymes, which are basically biological catalysts. Remember what a catalyst is? Catalyst is a molecule or an atom or a material that helps a reaction happen. It allows it to take place more easily. How that works, we're going to discuss in detail later. But a catalyst in chemistry is something that makes something happen, and it doesn't get involved in the reaction. It's almost like whenever it's around, the reaction happens faster. And when it's not around, the reaction doesn't happen very fast at all. So it's a helper. And we're going to see how it helps later. But it turns out that proteins are super helpers of biological reactions. In fact, almost every biochemical reaction that happens in your body, every digestion, every synthesis or building of molecules involves at least one or many enzymes that help it along. So proteins are very numerous and very important in this way. They can also function as messenger molecules. These messenger molecules are called hormones, and as we will see again later how they work, but essentially what they do is they allow different parts of a complex organism to communicate, right? So one particular organ or tissue can secrete, or can make these proteins, and then can secrete them into the bloodstream. And when they float around to other parts of the body, they interact with the cells because of the pushing and pulling, and they can cause other things to happen. So they form messenger molecules or communicative kinds of molecules. So you can see how very important these things are. An example of a cool protein is, just to give you an example, is hemoglobin. You've heard of that? Hemoglobin is a complex chain. We have a picture of hemoglobin here, but if we looked on the internet, we could probably find one. Just have a real quick look here. Let's go to Google and let's look up hemoglobin model. See what happens. Images, and there it is. You can see in these pictures. Trying to find a really good one that you can see in it. This is probably the best one. It's not as twisty and turny. It probably looks more like this, and you can see all the twisty, turny bits and pieces. But here what you have are, you can see there are 4 different colors, which means this is a quaternary structure of 4 different tertiary twisted knots all linked together. 4 of them, not just 2. You'll also see that there are little bits of iron in the middle of these blobs. Iron is a metal, and it forms ions, right? Which ions have lots of charge, positive charge. So that positive charge is attracted to bits and pieces of the polar R groups on the chains, which causes them to sort of surround it. So in the center are these little iron molecules, right? Like that. And the shape of this hemoglobin molecule is such that when oxygen gas is around, it's attracted to it. Oxygen gas, because of the shape and the way this molecule is set up, oxygen gas will be sort of lightly attached to this molecule. It'll come along and say, oh, I like this. It fits good. There's a nice positive-y, negative-y environment that fits my positive-y, negative-y environment, and we're just a good match, and I'll just hang out attracted there, right? And so inside your red blood cells, these proteins carry oxygen. So when the oxygen comes into your lungs, it gets picked up by these molecules. Now, these molecules can then let go of the oxygen once they reach your cells and trade off by also picking up a carbon dioxide, which then they can carry back to the lungs. So they're like a little taxi service, but their cool shape and their structure and the iron inside makes them attractive to oxygen molecules and carbon dioxide molecules, and they happen to be like a taxi for them. Incidentally, carbon monoxide molecules are also attracted to this. Carbon monoxide is a different shape and has a different attraction. So much so that if carbon monoxide attaches to a hemoglobin, it stays there forever and never gets out. It's like somebody taking a taxi and never getting out. That taxi is no longer useful to anyone else. And so what happens is that stays there until this hemoglobin is eventually broken down by probably the liver, I think. And the carbon monoxide essentially uses up all of your oxygen taxis. And if you have too much of it in your system, then you obviously essentially suffocate, because you might be breathing oxygen, but you have no way of getting it to your cells. So carbon monoxide is a very dangerous gas, and it comes from incomplete combustion of hydrocarbons, particularly cars and things like that. And that's how it works. It gets lodged in there so tightly that it can't come out. So there's an example of a protein picture. I can't do that. I have my pointer. I'll do it with the computer. If we just have another little look-see around here. This is a pretty good picture showing you the twisty spirals. You can see the secondary structure, the spirally, and then how the spiral is all knotted, the tertiary structure, and then the quaternary structure. Okay? They're calling these heme groups. That's where the iron is found. That's just something beyond what we have to look at. But it gives you just an example of the protein. So we're going to stop there. We're going to say that was a wonderful lesson on proteins. And next time, a little bit later on, we're going to come back to proteins and see how your DNA builds them. Your DNA actually has the instructions, which essentially, it's the instructions for which amino acids to put in which place so that you can get the protein that you want.